Hello, people. Anyways, I've noticed that this coronavirus or COVID 19 outbreak has been very serious, and I was going to point out that there might be potential solutions. However, I'm not an expert, but I believe that with my background in biology, I think that there could be certain ways in order to solve this pandemic. So, during my bioinformatics internship, I had to use something called Constance Information Miner software to detect endogenous ligands or metabolites within a malaria parasite called Plasmodium falciparum. This was used to identify fairly chemically similar ligands that could be similar to a list of 400 known anti-malarial compounds. I wasn't able to identify any drug targets because of my lack of experience and also just the limitations of the software. However, I'm wondering if maybe the same principle could be applied to trying to fend off this coronavirus. Currently, scientists are using the world's fastest supercomputer, including some of NASA's supercomputers, to identify novel drug targets against the coronavirus. They've analyzed the genetic sequence responsible for the structure of the receptor of the virus, which can be used to attach itself to host or human cells. So far, they have identified about 77 compounds that could be likely candidates. However, they're still trying to figure out which of these would be most effective at blocking off the COVID 19 receptor. Now, one thing I should also point out too is that if you notice, some people are immune to HIV. The reason why is because they lack something called a CCR5 receptor. So they have been analyzing the genomes of certain coronavirus patients, and they have discovered that there is a gene here called ACE2, which shows that depending on which variation of that gene you have, some people would have very severe coronavirus symptoms and others would remain asymptomatic. Um, however, I think that there might be a way where we could use something called CRISPR, which is a genetic tool that can splice DNA at certain points or make certain fragments and alter the genetic code. So I'm wondering if you could have people that show mild symptoms or asymptomatic to coronavirus, and you're able to take a fragment of their DNA, put it into like a plasmid or bacteria, breed the plasmid, and then perhaps maybe reinsert it into people where their variant of the AC2 would show severe symptoms. Now, the problem with CRISPR is that it's a new technology. It does have risks. However, these drastic times call for drastic measures. And I believe that maybe CRISPR could be used in order to alter, genomes of the virus or people in order to help reduce symptoms or help prevent such a fast spread of infection. I'm also going to point out here, though, that coronavirus is not a DNA virus, it's an RNA virus. So, DNA is basically inside the nucleus of the cell, which is used for, it contains all genetic information. For DNA to become a protein, it has to go through RNA. RNA is basically the medium that comes out into the cell. It fuses with ribosomes, and basically the genetic code comes from the DNA into the RNA, and ribosomes read that genetic information that codes for the proteins, and then proteins are made generally from guide messenger RNAs. So the coronavirus that we have here, the SARS-CoV-2 virus, is an RNA virus. So CRISPR-Cas9 is used in order to alter DNA. However, they've recently invented something called CRISPR-Cas13. This targets binding sites on RNA molecules. So what I'm wondering is, could it be used to modify the RNA inside the virus in order to render it useless? So I think that they could go into like an RNA sequence of the coronavirus, figure out which part of the genetic information is used to code for the structure or the function of the receptor that binds to human cells, and then basically create a prototype coronavirus that has a useless receptor. If that was to be reintroduced into the general public, perhaps that virus could end up reproducing and perhaps spreading its non-effective receptor onto others. And I'm hoping that maybe within 2 to 3 months, if a prototype virus like that was created, it could be used to help, you know, if its genetic information eventually would be exchanged with other viruses out in the wild there, that perhaps most of these viruses would eventually end up getting quite ineffective receptors as a result of that change in genetic sequencing. Anyways, I hope that perhaps my studying or my research could be useful for any scientists out there, but please let me know. Please share this video. I'm just doing this to help, the good of humanity. And, I don't know. I could be wrong. I could be right. I don't know. But the point is, this is a time of emergency. And I think doing anything right now that could help prevent the spread of coronavirus would be useful. Anyways, thank you. And I hope you shared this video.